Very good evening to everyone. Today we will discuss about how to make a research proposal. Usually for a MSc or PhD students, they face problem in making research proposal. When you are going to take admission in MPhil, in PhD, okay, you have to present a research proposal. So today we will discuss with a case study and I selected the case study as a evaluation of ethnopharmacological values of carnivorous plants of Odisha. So this is a one research proposal. So what will be the segment, what it will run, okay, we will discuss it. So as per the title was ethnopharmacological values of a carnivorous plant. So first we will discuss about review of literature. So you can observe, okay, from the review of literatures, okay, we have collected some data and we have to present the data not in written form. Usually what we do, we write lot, but we have to write less, we have to present more. So here is the distribution of carnivorous plant like in world, India, or Odisha, and diversity of five most reported genera of carnivorous plants. So you can observe here Foeticularia, Drosera, Nepenthes, Peningula, Genelisa. So it should be the representation should be in tabular form, in graph form, not in word only. Now, rationale of the study. What is the background of the study? Why you have selected the topic? Like we have written, carnivorous plant, Drosera species are magical plant groups having different types of bioactive compounds and enzymes. They have a traditional therapeutic values and used throughout the world, but still they are unexplored. They are used against respiratory problems. Nowadays, we all are facing the problem related to respiratory diseases. Hence, to study the ethnopharmacological values of Drosera species could give some herbal or pharmacological agent to reduce the problem related to respiratory disease. So these are the rational background of the title or the study. And also you can put some photograph like I have put Drosera indica, Drosera peltata and Drosera burmani. Now objectives. We have to write the objective. Like I have written the objectives for this title. First objective, survey to document the distribution and ethnomedicinal qualitative and quantitative values of Drosera species in Odisha. Collection of plant specimen and herbarium submission for authentication of a species for further experimental works. Qualitative phytochemical analysis of bioactive compounds present in Drosera species found in Odisha. Qualitative analysis of primary metabolites present in Drosera species reported from Odisha to carry out the antimicrobial activity of Drosera species against pathogenic bacteria responsible for respiratory disease, to test the toxicity of the experimental plants to Artemia salina, analysis of different enzymatic activity present in Drosera species, isolation, purification, and characterization of antibacterial agents from Drosera species found in Odisha. Now research questions. So in proposals here we have to write the research question or hypothesis. Like is respiratory disease a critical problem? Question mark. Should we find the herbal or pharmacological agent from plant wealth from carnivorous plant? Question mark. Is there any study on distribution of carnivorous plant? Is there any works reported on the phytochemical constituents? Is it need to correlate the bioactive compounds, pharmacological values, and enzymatic activities of carnivorous plant? So these are the research questions. This is also a most important part when we present our research proposal for MPhil, MSc, and PhD. Now the methodology. It should be in brief always. Like I have written for this topic, selection, identification and enumeration of selected experimental plants from a study area, collection of experimental plant species, preparation of plant extract, phytochemical assays, qualitative test for primary metabolite, antimicrobial activity of Drosera species against pathogenic bacteria responsible for respiratory disease, cytotoxicity test using brine, shrimp or Artemia salina, test for enzyme activities, isolation, purification and characterization of active compound. Now, this is the methodology in illustration form. You can observe it. This is the best way. Okay, never forget to write about the source or the reference, which standard protocols you are going to use. You can observe I have written like in the part of antimicrobial activity. This diffusion essay, Sarkozy et al. 2007. Agarwal diffusion essay, Alien et al. 1991. Broad dilution essay, Rai et al. 2010. So the source should be given and it should be in illustration form, not in writing form. That will be a lengthy. So it will be a what that means comfortable for both a student or examiner. So they can understand easily what you want to say, what you want to explain. So that will be a better or the selection rate will be high. Now expected outcome. This is the another important part. Like distribution and ethnomedicinal values of Drosera species in Odisha will be documented. Qualitative phytochemical analysis of bioactive compounds present in the whole plant of Drosera species will be evaluated for proving its pharmacological values. Quantitative analysis of primary and secondary metabolites present in Drosera species will be evaluated. Antimicrobial activity of Drosera species will be documented. Toxicity of the experimental plant to Artemia salina will be evaluated, which make it preventive agent against viral infection. Analysis of different enzymatic activity present in Drosera species will provide a base data for further work on medicinal enzymes. The isolation, purification, and characterization of new or derived compounds will be documented having antibacterial activity for formulation of future drugs. Thank you very much. Now you can understand how we will write or present a research proposal for MSc or PhD. Thank you very much.